Hi everybody, this is Digital Embrace. I'm going to show you a few tricks for working in our new Ribosome Evolution lab, where we are trying to find a ribosome that will process synthetic amino acids that are called cyclic beta amino acids that researchers want to use to make new exciting synthetic materials. Now, in our previous lab, the ribosome optimization, we were using the wild type sequence, the E. coli, that they've been using in the laboratory, but it hasn't worked really great. And so they wanted us to find a more stable ribosome that could more reliably produce more protein. And so we found one that produces 40% more protein, and that's what we're using for our starting basis in this lab. Now I've pulled up a design here from Bugakman, and you can see where he said that he's going to be working on the P loop here. When you first pull it up, somebody else's design, it's kind of hard to see where they've made the mutations. Where is it here now? I can see there's the purple. So I'll zoom in on that. And another way you can look for these is to go into the browser, designs here. And if you move over, that's too far, back a little too far again, there we go. It says library nucleotides. So you can see which nucleotides have been chosen for each design. Okay, I'm going to zoom in some more here on this design that we're looking at to look at the library that Bugakman created. And it's here in the middle. I'm going to zoom in one more time, make it a little easier to see. So he has chosen 4 positions and mutated them, and this is in the P loop. And the way you choose them is you click the dice tool, click on the nucleotide, and there you go. You can also deselect the same way. Now, after you've made your library, say here he has 4, he has also mutated all 4 to the sequence he wants tested. So there's 2 actual experiments that are going to happen. There's the big, really exciting new experiment where Antje will be taking our library and testing all possible combinations, testing all possible sequences within that library group. She's also going to do a separate ISAT where just a standard ISAT experiment where she'll test the sequence that you indicate here with your mutations. So be sure to indicate which sequence you believe will be the best among all possible combinations that are tested in the primary experiment. So you see all of them that he chose here are IUPAC violations. And that is totally fine. That's actually great because they're all being tested. All possible combinations, all nucleotides will be tested. So there's absolutely no issue with that at all. But we left it in there so people just would have that knowledge there in case they wanted to know something about, well, what have we been testing previously, what positions. And then the flags also left in here so players can see what flags come up. So on this one, it's the 2252. Of course, it says P loop because it's the P loop. And it's a tNRA contact, which is very interesting because the tNRA is what brings the amino acid into the PTC, into the ribosome. And it's at the P loop actually where it then gets processed and added to the peptide chain. So this would be an interesting place to test. So given another example, is if I mutate this G here to A, first of all, it's going to tell me, hey, you haven't added that to your randomized positions. So we go to our dice, say I wanted to make this design and add it, and now it's part of my randomized positions. And the flag's telling me it's a tRNA contact. Well, that's really interesting, isn't it? And it's also, it pairs with base 2275. So that doesn't show up in our 2D display. So when that's a tertiary interaction, we call it 3D sometimes, a tertiary interaction that exists in the actual ribosome when it folds up. So 2255 generally bonds with 2275. So if I were going to make a design testing that position, I would probably also test 2275. So I'm going to mark that also. Here's 2275. And that was a C. So since I mutated this to A, I would probably then choose to make this a U. Okay, well, if you have any Further questions, please post in the forum or read in the forum, our excellent forum. Lots of different ideas people will be posting. If you haven't joined the forum yet, go to our homepage and under community, here's the forum. So click there and sign up. You may have to create another password. You can use the same password as you have already for Eterna. It'll work fine. All right, and I look forward to seeing the designs everybody comes up with. This is a super exciting lab. All right, I'm out.